My name is Sajni, and my topic is on the role of glycoproteins in cell recognition and adhesion. So, glycoproteins are carbohydrates that are attached to many different proteins. Many glycoproteins are part of the cell membrane. Their roles involve processes such as cell recognition and cell adhesion. During cell recognition, glycoproteins help recognize each other due to their unique pattern of glycosaccharides on the cell surface that are read by corresponding glycoproteins on another cell. Glycoproteins will often bind together if they are matched. Another type of glycoproteins are known as cadherins that require extracellular calcium ions to function and are required for development. E-cadherin is epithelial tissue and cadherin is nervous tissue. P-cadherin is our placental tissue. They act to drive the adhesion of cells of a particular tissue type. So NCAMs are known as neural cell adhesion molecules, and they are large plasma membrane glycoproteins. When NCAMs and cadherins, they're both are carbohydrate groups that determine the strength and specificity of cell-to-cell recognition and adhesion. Shown in this image are NCAMs and the E-cadherin and the glycoproteins. NCAMs have repeating chains of negatively charged salicylic acid, which changes during development. If there are vesicles with NCAMs with little salicylic acid, they bind tighter than those with large amounts. If there's a loss of salicylic acid from a glycoprotein, it may target old erythrocytes for destruction in the spleen. The enzyme neuraminidase can cleave the terminal salicylic acid group as a mechanism to identify old red blood cells for retirement. So during inflammation, leukocytes initiate attachment to the endothelial cell surface through the selectins and then they stabilize the adhesion through the interaction of an integrin and an ICAM. So this is a structure of the glycoproteins. So in this image, as you can see, there are many proteins that are floating around either in or around the membrane of the cells. They move and are able to interact with the cell's environment. Glycoproteins are always found on the extracellular, the outside of the plasma membrane with the sugar facing out. In this image, there are various proteins such as the protein channel, the transport proteins, the peripheral proteins, the integral protein, also known as the gallbladder protein, and the surface protein. So an important aspect of glycoproteins is they help our immune system. They're able to recognize foreign invaders and defend our body against them. When embryonic tissue is exposed to antibodies that interact with NCAMs, the cells do not bind to each other and neural tissue is not favored. Glycoproteins in the immune system, their function is influenced by the process known as glycosylation. Glycosylation has the ability to modify cell function since it is able to modify proteins. Glycosylation influences the function of all immune cells. Glucans play an important role in intracellular contacts and leukocyte migration, which are important in activation and proliferation of leukocytes and during an immune response. The main immune proteins involved are the TCR, MHC, TLR, and antibodies are glycosylated. Sugars on the surface of pathogens and cell surface glycoproteins are recognized by carbohydrate binding proteins called lectins. Changes of glycan structure are common in many pathological processes occurring in the immune system and are therefore used as molecular markers of different diseases. One type of such a disease is known as breast cancer. So, breast glycoproteins at an early stage is predicted to predict disease development and the progression. As well, they could facilitate the development of effective therapies. So during glycosylation, there are two key players known as mucin 1. Mucin 1 is a large transmembrane protein. A change in glycosylation is due to the exposure of the protein core that mediates the attachment of cells to tissues at different sites. This is important in the formation of remote metastasis. So if there's underglycolation of mucin 1, it is associated with a higher grading and can serve as a diagnostic marker. Another key player is known as galectins, which are indigenous galactoside, and they are binding lectins on the binding site and are involved in cell adhesion and growth regulation. If galectin 3 is associated with mucin 1, this binding results in what is known as a fridin-rich antigen, which promotes cancer cell adhesion to the endothelium and will contribute to metastasis formation. So the diagram shown here is showing how the galectin 3 and the mucin 1 interact, and together they promote cancer cell to cell and cancer cell endothelial adhesion and metastasis. The alteration in the adhesion properties of the cancer cells and the extracellular matrix will result in the invasion of epithelial cancer cells through the extracellular matrix. So letter A is showing the intravasation. Letter B is showing the primary tumor sites. Mucin 1 is being overexpressed on the cell surface, preventing the adhesion of the invaded cells with adjacent cells, and is being polarized because of the response to the binding of the galectin 3 that is circulating, which is inducing the exposure of the smaller cell surface adhesion molecules or ligands, and the cell becomes sticky. So letter C, the sticky cancer cells are adhering to blood vascular endothelium. Letter D is showing that they stick together to form a microtumor. Letter E is showing that they are able to bind to the endothelial cells, which both will increase cancer cell metastasis to distinct organs. So a recent finding I found, the experiment was they used a lectin enrichment technique to evaluate changes in glycoproteins in the blood serum of patients who were diagnosed with ductal carcinoma in situ and invasive. So this enrichment was applied using CON-A, ALE, and WGA to capture MAN, GAL, and GLC, NAC-sialic acid residues. So using this, they found that fibrogen beta change and neuropilin 1 were downregulated in ductal carcinoma and was not found in the invasive breast cancer. So the association of lipid transport and metabolism were related to tumor growth and metastasis. So my question is, what are ways in which these glycoproteins have the ability to detect the process of disease development and cancer? Thank you for watching.